Hello everyone. Thank you for choosing to attend this workshop. My name is Janine Colling and I'm the manager of the Vibrational Spectroscopy Unit at the Central Analytical Facility. I hope you found this training to introduce NIR spectroscopy and multivariate data analysis useful. During the training, we have tried to explain some of the concepts for someone who is at a beginner's level to establish a good foundation to build on. Once you've finished watching the videos, you are welcome to send us any questions and suggestions you may have via email using the indicated email address. A summary of all the questions and answers will be emailed to everyone. Thank you. If you're on Facebook, please go like our Facebook page to receive the latest information, for example, about new training opportunities. During the training, we are going to focus on providing an overview of the following two sections. In the first video, you will learn more about NIR spectroscopy and hyperspectral imaging to understand how this technique works and what type of data is generated. In the next two videos, we will introduce multivariate data analysis, which will look at principal component analysis and developing classification and quantification models. This will only serve as an introduction to these techniques. To understand what near-infrared means, we will first go over the basic principles of light. Then we will discuss what you can learn by measuring light. Next, we will investigate the differences between conventional spectroscopy and hyperspectral imaging and look at the types of hyperspectral imaging equipment in the unit. After having a more clear understanding of this technique, we will briefly mention some of the applications of NIR spectroscopy. We will finalize presentation by discussing the advantages and disadvantages of this technique. So let's start by looking at light. Light, which is visible to your eyes, is actually electromagnetic radiation and can be described as a wave that is moving through time and space. This wave has both an electric and magnetic field and carries electromagnetic energy. Waves can be characterized by their frequency, which is the number of times a wave passes by a certain point in a given unit of time. And the unit for frequency is Hertz. A wave can also be commonly expressed as wavelength, which is the distance required for a wave to complete a full cycle, and the units are expressed in nanometers. Waves can also be expressed based on the amount of energy that they contain, and the units for photon energy is expressed in electron volts. All of these are units for electromagnetic radiation. The electromagnetic spectrum is made up of waves of different frequency or wavelengths. The spectrum covers a range starting from those with shorter wavelengths such as the gamma rays to those with longer wavelengths such as the radio waves. Shorter wavelengths have higher energy, whilst the longer frequencies have less energy. In visible and near-infrared light, we are especially interested in the visible and infrared part of the spectrum. Visible light are those wave bands from 400 to 780 nanometers, whilst infrared light are the wave bands from 780 to 1 million nanometers, and it can be divided into the near, mid, and far infrared light. We're interested in the near-infrared radiation, which are those wave bands from 780 to 2500 nanometers. As mentioned in the previous slide, radiation contains energy. This property has made electromagnetic radiation useful for several of the activities we perform in our daily lives, including switching on the light when it gets dark, listening to the radio, heating our food in the microwave, recording people's temperature, various medical applications, and generating electricity. To understand how this is possible, we have to take a closer look at what happens when the energy in radiation is absorbed. When radiation interacts with matter, the energy captured in the wave bands can be absorbed by the molecules in the sample. Since different wave bands carry different amount of energy, they result in various molecular responses as indicated. Visible light results in electronic transitions, whilst infrared light results in molecular vibrations. Over the years, different instruments have been developed to measure these responses. Measuring the interaction of radiation with matter is called spectroscopy. A little bit more about near-infrared light. NIR light was discovered by the German-born British astronomer Sir William Herschel. He discovered infrared light through using a basic experiment which involved splitting sunlight using a glass prism and shining the light on bulbs which contained thermometers. By doing so, he noted that the radiation above red light resulted in an increase in the temperature. He called this region of radiation the infrared region. So now that we know more about what infrared light and radiation is, let's move on to spectroscopy. Spectroscopy measures the interaction of light with matter. During spectroscopy, there is usually a source of light, such as the sun, which provides the radiation. When the sun shines on a sample such as the leaf, a part of the energy in the radiation is absorbed by the leaf. Some of the radiation which is not absorbed can pass through the leaf and be transmitted, or it can be reflected. Spectrometers can be positioned to measure the amount of light transmitted or reflected, and the amount of light absorbed can be calculated. This information can be represented as a spectrum, where the amount of radiation reflected, absorbed, or transmitted over the wavelength range can be represented. So what can we learn from the spectrum? A classical example which a lot of people are familiar with is UV spectroscopy. This technique can be used to quantify a compound in an extract prepared from a sample. To use this technique, a standard solution with a known concentration of a compound, in this case nucleic acid, is first analyzed. This sample can be placed in the instrument and the amount of light transmitted calculated. From the percentage light transmitted, the absorbance can be determined. According to the Beer-Lambert law, there is a linear relationship between the absorbance and the concentration of the molecule. So the more of the compound there is, the higher the absorbance. From here, there are two ways that the concentration of a compound in a new solution can be determined. For the first method, the molar extinction coefficient can be calculated using the standard solution of the compound. The extinction coefficient is an indication of how strongly a chemical attenuates light at a given wavelength. It's an intrinsic property of the chemical. Once this is known, the absorbance at the particular wavelength can be determined for a new solution and the concentration of the compound determined by rearranging the formula. The second method involves preparing a standard curve using different concentrations of the compound of interest. Since there is a linear relationship between the concentration and the absorbance, an equation describing this relationship can be determined. The equation can be used to determine the concentration of unknown samples from its absorbance. One important thing to note with UV spectroscopy is that to determine the concentration of a compound in a new sample, the compound has to be extracted from the sample to obtain it in a solution that can be analyzed. This is an important difference with NIR spectroscopy. During NIR spectroscopy, the sample is first scanned to obtain an absorption spectrum. This means that the spectrum contains all of the chemical information of the sample, including the compound of interest. Next, several samples are scanned, and then the samples are analyzed using normal reference methods to measure the compounds. Ideally, various samples containing a range of concentrations that can naturally be present in the sample should be analyzed. Mathematical modeling is then used to link the concentration to the spectral information. The data is first split into a training and a test set, and the training set is used to teach the model to predict the property. Once the model is developed, a test set is used to determine how accurately the model predicts the values. When a good model exists, used samples only have to be scanned and the model applied to predict the quantity of the compound. NIR spectroscopy can be used to quantify compounds in solid and liquid samples. When using solid samples, the distribution of the compound in the sample should be taken into consideration. Another application of NIR spectroscopy on solid samples is to study differences in physical properties. This is possible because differences in particle size result in light scattering. In the image on the left, the sample has smaller particles. This allows for less transmittance of light and higher scattering and reflection of light. In the image on the right, the sample has larger particle sizes. Here, there is less scattering, more transmission, but lower reflection of light. If you look at the spectrum, you will see that there is a baseline shift, which is likely caused by the difference in scattering within the sample, with the smaller particle sizes resulting in a higher spectrum. This is an important point to keep in mind when selecting samples to use and when performing analysis. In some instances, it may be a goal to study differences in particle size, whilst in other instances, it may be of value to grind the samples to the same particle size to exclude this variation. When performing chemical analysis using NIR spectroscopy, the molecular bonds such as CH, NH, OH, and SH, which occur in organic samples, can be analyzed. These bonds act like springs, and they start to vibrate at specific wave bands which match the energy required to start vibrating. There are different types of vibrations, for example, stretching, scissoring, rocking, wagging, and twisting. By looking at the frequency where a chemical bond absorbs, we can learn more about the identity of the molecule. And by looking at the absorbance level, we can learn more about the amount of the compound in the sample. At this point, we understand that light can give us information about the concentration of a compound in a sample and provide information about particle sizes. But if all compounds can interact with radiation and absorb energy, how can we get information about specific compounds? Let's start again with UV spectroscopy. We saw that with UV spectroscopy, there is electronic transitions, which means that the electrons move to higher electronic states. The energy difference between the two states is called the band gap. The energy of a photon must exactly match the band gap for the photons to be absorbed. Thus, molecules with different chemical structures have different energy band gaps and different absorption spectra. The difference in infrared light is that the energy is not enough to excite the electron to a higher state. Instead, the energy causes vibrational excitation of covalently bonded atoms, resulting in vibrational transitions. Just as the energy required to excite an electron in a particular atom is fixed, the energy required to change the vibrational state of a particular chemical bond is also fixed. When the transition is from the ground state to the first vibrational level, we call this transition the fundamental transition. And when it is from the ground to the second, third, or fourth level, we call these overtones. You will also hear mention of combination bands. These occur when two or more fundamental vibrations are excited at the same time. It's important to note here that the overtone bands are weaker bands and that they occur in the NIR range, whilst the fundamental transitions occur in the mid-infrared region. In the past, several instruments were developed which operated in the mid-infrared region, as specific chemical bonds could be ascribed to the peaks. In contrast, NIR spectroscopy was neglected for a long time, as very little information in that spectral region could be deduced due to the broad, superimposed, and weak absorption bands, which made it difficult to interpret. With the advance in multivariate data analysis tools, it became easier to analyze the spectra. By applying these tools, certain chemical bonds could be ascribed to the various absorption regions, and tables such as these were generated where specific chemical bonds were ascribed to the combination bands and overtones. This allowed the spectra to be used for exploratory and modeling purposes. At this point, let's summarize what we've learned thus far. Light is actually electromagnetic radiation, which contains energy and forms an electromagnetic spectrum. The energy in radiation can be absorbed by molecules and result in various responses. Molecules with specific chemical bonds in organic samples absorb energy in NIR radiation and start to vibrate. Spectroscopy measures the interaction of radiation with matter and records the response as a spectrum. In NIR spectroscopy, the chemical bonds are linked to overtones and combination bands. NIR spectroscopy can be used to study the chemical and physical properties of samples and can be applied to liquid and solid samples. Analysis of the results requires the use of multivariate data analysis tools. Next, we're going to look at conventional spectroscopy instruments versus hyperspectral imaging. Here you can see many different types of conventional spectroscopy instruments. They offer many different ways to obtain the spectra of the sample depending on whether it's a liquid or a solid. Examples include presenting the sample in a sample cup, using a fiber probe, a cuvette for liquid samples, or just holding the instrument in contact with the sample to record the spectra. The sample is usually presented to the instrument in the way that you want to perform a routine analysis. When a conventional spectroscopy instrument is used, a representative spectrum of the sample is obtained. This is not a problem if the sample is homogeneous, such as liquids, but what about samples where the chemicals are not evenly distributed? To obtain a representative spectra, the sample can be scanned several times and all the spectra averaged, and this is called object-wise analysis. In some instances, it may however be interesting to see how the chemicals are distributed within the sample. To achieve this, a hyperspectral imaging camera can be used. This involves placing the sample on a sample stage which moves the sample past the camera and captures an image of the sample. Every image consists out of pixels, and in a normal color image, the absorption at 3 wave bands associated with red, green and blue is measured for every pixel. When using a multispectral camera, the absorbance at a smaller number of wave bands can be determined. In hyperspectral imaging, the absorbance over a range of wave bands is recorded for every pixel in the image. By doing this, we can compare the absorption spectra for every pixel in the image. In the example shown here, an apple was bruised. In the normal color image on the left, the bruising was not necessarily visible. Since bruising may result in chemical changes and changes in the physical properties of the area, this information can be captured in the absorption spectra. By creating an image comparing all of the absorption spectra of the pixels, it becomes possible to detect these differences. The data obtained with hyperspectral imaging is a 3D dataset called a hypercube. The first 2 dimensions represent the pixel position within the image. The third dimension represents the absorption information over the wavelength range. Since hyperspectral imaging captures images, the distribution of a compound can be studied. However, it is important to think about the way that you will present the sample to study the distribution patterns. There are 3 different ways of performing imaging. In the lab, we have a push broom system. This system scans line by line. A whisk broom system scans point by point and therefore records the information more slowly but generates more accurate results. The steering mode records the whole image in a snapshot and creates less informative information. The data is usually recorded in reflectance mode, and depending on the field you are working in, it can also be converted to absorbance. The 2 hyperspectral imaging systems which are available in the vibrational spectroscopy unit include the visible near-infrared camera, or NIR, and the shortwave infrared system, or SWIR. The NIR collects information in the 400 to 1000 nanometer spectral range, and this includes the visible and NIR region. The SWIR operates in the 950 to 2500 nanometer range. Every image in the NIR has 1800 pixels in a line and 384 for the SWIR. The spectral resolution for the NIR is 3.29 nanometers, and for the SWIR it's 5.45 nanometers. This results in 182 bands for the veneer and 288 wave bands for the sphere. Prior to imaging, the sphere camera has to cool down to 150 Kelvin. The veneer camera has a CMOS detector and the sphere camera an MCT sensor. These are the main differences between the two cameras. In the unit, there are 3 different types of hyperspectral imaging systems, and the difference is that 2 of the systems only have a single camera setup. There is also 1 double camera setup. Here you can see each of the cameras which record the hyperspectral image. The sample is placed on a translation stage, which moves it past the camera. The sample is illuminated by 2 halogen tungsten lights, which provide the light in the spectral range we are working with. Each camera is equipped with 3 different lenses, which determine the field of view. The field of view means how much of the stage the camera sees and also determines how large every pixel is. An example with a microscope lens is that you will be able to see wheat seeds clearly, but any resolution smaller than that is not possible. During imaging, a 50% gray reference is used as a white reference. The shutter is also closed to collect a dark reference. Both of these references are used to correct for differences in illumination over the field of view. So it's used to correct the raw image to obtain the reflectance images using the formula as indicated. The Breeze program from Predictera is used to operate the cameras and to record the images. Once you decide to use the hyperspectral cameras in the unit, you will receive training on how to use the software and set up the cameras for imaging. Now let's take a look at what we can do with NIR spectroscopy and what you could use it for. NIR spectroscopy is often described as a secondary analysis technique. The reason for this is that it heavily relies on the information that you already know about the sample, and it's based on information derived using other techniques. There are 2 types of applications. One can either use NIR spectroscopy for exploratory analysis or for developing models. Both of these are based on the chemical and physical differences between the samples. Let's look at an example of the application of exploratory analysis. There's a wide range of fields where this technique can be applied. An example in the pharmaceutical industry is to evaluate whether NIR spectroscopy can distinguish fake tablets which look like the real product, or to distinguish between products. In the photo, there are several tablets which look the same. Some of these contain different chemicals or active ingredients, whilst one is not a real tablet. When we analyze NIR spectroscopy data and perform multivariate data analysis, we can evaluate whether this technique can distinguish between the tablets based on their chemical differences. In the image, the dots represent the pixels related to the different tablets in the image. You can observe that the pixels with similar properties group together. The reason for this is because the pixels have the same properties, in this case the active ingredient. For example, this cluster belongs to the tablets containing paracetamol, and this cluster to those tablets containing acetyl salicylic acid. We can also identify the tablets which contain ascorbic acid and the fake product. Exploratory analysis therefore allows us to compare the chemistry of samples and to investigate if the chemical differences result in meaningful information or applications. Other examples include the agricultural industry, where a farmer may be interested to determine if the technique can be used to detect diseased plants. If the disease results in a consistent chemical or physical change, it may be possible to identify the diseased plants. In the food sector, fruit may be classed into different grades or levels of maturity. If the grades or maturity levels are based on consistent chemical differences between the categories, then the application of this technique to distinguish between them can be investigated. This might also be useful to grade other food items. A final example is in the wine industry, where there may be interest to evaluate if wine originating from different parts of the world may be chemically different due to the area where the grapes were cultivated. These chemical patterns act as chemical signatures, and it may be useful to determine if spectroscopy can pick up these chemical differences to determine the origin of the wine. Exploratory analysis therefore allows us to evaluate if we see certain patterns or trends in the data which are created by differences in their chemical or physical properties. When performing exploratory analysis, there are some important considerations to keep in mind. Exploratory analysis can be used to study the differences between samples, and these differences are usually based on the chemical and physical properties of the sample. NIR spectroscopy can be used to detect chemicals which are present in ppm levels, but this technique is therefore not useful to study compounds which are present in trace levels. When preparing samples, keep in mind that differences in particle size is captured in the spectra. In some instances, it may be of interest to study particle size differences. For example, in these pictures, the hardness of maize seeds were evaluated, and areas with different hardness levels could be identified in the PCA scatter plots as these pixels cluster together. In other instances, it may be of value to grind the samples to the same particle size to exclude this variation. A final consideration is that water absorbs strongly in NIR. If there are differences in the moisture content, this will be very prevalent during the data analysis, whereas differences in other chemicals may only appear to be minor. NIR spectroscopy can also be used to develop models. There are two types of models: classification models and quantification models. Classification models are developed when a category is assigned to a sample. In the agricultural industry, the goal may be to determine if a plant is healthy or diseased. In the pharmaceutical industry, the goal may be to develop a model which can determine if a tablet is real or fake. In the food industry, the goal may be to develop a model that can determine if the wine comes from Europe or South Africa. Each of these classes therefore represent a category to which a sample may be assigned. In quantitative models, the chemical content of a sample is predicted. To develop this kind of model, representative samples need to be obtained, which include all of the sources of variation that may result in chemical differences. For example, for wheat, there may be chemical differences between the varieties, years of collection, or area where the wheat was cultivated. It is also necessary to obtain samples which span the range of the compound for which the calibration has to be developed. To develop quantitative models, there are some important considerations, some which were already mentioned on the previous slides. These include taking into account all the sources of variation in samples for which the model will be used and collecting representative samples. The natural range in which the compounds occur in samples has to be established, and samples spanning this range for each of the sources of variation should be collected. It will also be necessary to have a good reference method to obtain the chemical values for the samples. A model will only be as good as the values that you presented. If your reference method is therefore not accurate, one cannot expect the model to predict the values more accurately. The second last point is that if the model will be applied to quantify compounds in fresh samples, but the reference method determines the values on a dry weight basis, it will be necessary to convert the values used in the model to the fresh weight basis. Finally, the type of sample and the stage of the process where the model will be applied should be determined, and all of these factors should be incorporated when developing the model. If at this point you're not sure if NIR spectroscopy is the right technique for you, you are more than welcome to contact us and discuss your idea. Expect that we will ask a lot of questions to learn more about the type of samples you are working with and what you're trying to achieve. It's also of value to think about the type of analysis that you want to do and what you expect from the results. To wrap things up, let's discuss some of the advantages and disadvantages of this technique. NIR spectroscopy is a rapid analysis technique. Once the model exists, the sample only has to be scanned to obtain the spectra, and the model applied to predict the property of interest. Analysis can be performed in real time, and it requires almost no sample preparation. The values can also be accurate depending on the reference values used to develop the model. This technique is a non-destructive technique, which means that the sample can be used for other purposes or reanalyzed over time. Since there are no chemical analysis, there are no chemicals or waste, and it therefore reduces the costs. A large number of samples can be analyzed at once, and if models to predict multiple properties exist, all of these measurements can be determined from scanning the same sample. Models can also be used to analyze new samples. There are a few disadvantages of NIR spectroscopy. Initially, it may be expensive and time-consuming to collect and analyze the samples to develop the models. NIR spectroscopy equipment is also expensive, but these are accessible through the unit. The analysis of the spectra may be complicated and differences in spectra may be subtle. Unfortunately, this technique is not useful for trace analysis. And finally, to develop quantitative models requires access to good reference methods. To finalize, let's briefly go over the points discussed. We said that light is actually radiation which forms part of the electromagnetic spectrum. Visible and infrared light is part of the spectrum and spans the indicated wavelength regions. Spectroscopy measures the interaction of light with matter. The interaction involves the absorption of energy and the reflectance and transmission of light. NIR spectroscopy measures changes in vibrations of chemicals with covalent bonds. The absorption of energy is recorded as a reflection or absorption spectrum. The difference between conventional and hyperspectral imaging is that conventional instruments record an average spectra for the sample, whilst hyperspectral imaging creates an image where the absorption data for every pixel in the image is collected. NIR spectroscopy can be used to study chemical and physical properties of samples. By applying multivariate data analysis, this technique can be used to explore differences in these properties or to develop models to predict concentrations or assign samples to categories. The main difference between this technique and UV spectroscopy is that with UV spectroscopy, the compound has to be extracted from the sample, whilst with NIR spectroscopy, the compound is measured in the sample. If you have any questions about the training presented here today, or any suggestions, please let us know by sending an email. We thank you for your time and we hope to see you in the unit.